All right, so I want to welcome everyone to our very first screencast for Chapter 6. And I say first screencast because we're going to take the information in Chapter 6 and actually break it down probably into two screencasts. One of the reasons we're going to do that is because we're going to start our discussion of the cell, more specifically the eukaryotic cell. And if you remember back to the information we had looked at in Honors Bio, the eukaryotic cell is considered a complex cell, which means it has lots and lots of different parts. Now, you were introduced to those parts, but what we're going to do in AP Bio is we're going to look at those parts in a little bit more detail. So before we get started talking about those different parts, what we want to do is we want to, of course, have the understanding, which most of us do, that the cell is considered the fundamental unit of life. And what that means to us is that any organism that is considered alive will be made up of cells. Now, the cell is considered the simplest collection of matter, which is considered alive, when you consider the hierarchy of biological organization. Now, that hierarchy was discussed back in Chapter 1, and what we mean by that is if you think about us, for example, you start at the organism level, and then you start working your way down. You think about the organ systems, then you think about the organs, you think about the tissues, and then, of course, you think about cells. Now, cells are considered the basic units of structure and function. Us being multicellular creatures, we're made up of lots of different types of cells. Now, those cells are a little bit different from each other because they each have a very unique structure that allows them to function specifically within our body. In other words, they do different jobs for us. Now, all cells are going to demonstrate most of the themes that we have introduced back in Chapter 1, and it's going to reflect the idea of emergent properties. If you take us and you basically pull us apart, again, we're lots of tissues, we're lots of different cells, but if you put us together, if you put all those parts together, we emerge as us, and that's what we mean by emergent properties. So all cells are going to be related by their descent from earlier cells. But as we had said in the previous two bullets, they have been modified because they have different jobs they need to do to make sure that we survive. Now, in order for us to make some observations of these different types of cells, of course, we're going to need a tool. And in science, we're going to use a microscope. So scientists are going to use microscopes to visualize cells that are too small to see with the naked eye. So most of the cells that you would encounter are going to be really, really super tiny. One of the microscopes that you would be exposed to in the classroom would be something called a light microscope. In fact, that's probably the only microscope you'll be exposed to because the other ones we're going to talk about really are beyond our ability to be able to purchase and use. This particular type of microscope is going to use visible light, and it's going to pass through the specimen that has been cut really thin, and then it's going to pass through a series of glass lenses, usually two. And the purpose of this is to magnify the image, which basically means to make it appear bigger. Now, the quality of that image is going to depend on three things. The first one is going to be the magnification power, in other words, the ratio of an object's image size, what we see, to its real size. The second is going to be the resolution. Now, the resolution is going to measure the clarity of the image, or the minimum distance of two distinguishable points within our image. And then we're going to be looking at contrast. So this is going to be the visible differences in parts of the sample. And so, of course, depending on the type of scope that you're using, these are going to change. And in a light microscope, you're going to be somewhat limited in what you could actually see. Now, over here on the right, this chart is just giving you an idea as to the size of cells. And if you look here, of course, we have humans at the very top. Then if you notice, most plant and animal cells are going to fall between the 10 micrometer and 100 micrometer range. When you go below 10 micrometers, then what you're looking at is you're probably going to be looking at some bacteria all the way down to 1 micrometer. Some of your smallest bacteria could actually be even smaller at 100 nanometers. Then, of course, you come down here to viruses and the different cell parts that we've looked at, and all the way down to the macromolecules that we've looked at, like the proteins and the lipids. Then down here at the very bottom, we have atoms, which are scaled at about 0.1 nanometers. So light microscopes, as I had said, are going to be very limited in what they could magnify effectively, and it's going to be limited to about 1,000 times the size of the actual specimen. Resolution is going to be limited to about 0.2 micrometers or 200 nanometers. and this is going to be represented by the bacteria that you could probably see using a light microscope. Now, historically, there are two people I want you to be aware of when it comes down to microscopes. Around 1665, we had somebody by the name of Robert Hooke, and he was the first person to actually observe a cell. And what he was doing is he was looking at the cork tissue. I think it was from a maple tree. And what he observed is he observed the cell wall. Now, this was cork, so these were non-living cells. As we know, the cell wall usually remains after a plant has died. Now, 1674 saw Anton van Leeuwenhoek, and he was the first person to actually see living organisms, microorganisms, through a very simple microscope. So various techniques are going to be used to enhance the contrast and actually enable cell components to be stained or labeled. This is something that you probably have done in science class. For example, I know in my honors classes we use iodine to stain cheek cells. And when we do that, that helps to bring out various cell parts, more specifically the nucleus of the cheek cell. And you can also see some of the cytoplasm within that cell as well. Now, most subcellular structures, including organelles, and this is the term you're familiar with, these are the membrane-enclosed compartments that you would find within the cell, are going to probably be too small to be resolved or seen by a light microscope. Again, we're kind of limited to what we can see at about 1,000 times. You could probably see the nucleus, you could see the nucleolus, you can see cell membranes, you can see the cytoplasm, but a lot of the other organelles are going to be too tiny to see with this type of scope. So as I had sort of hinted to maybe a little bit earlier, the light microscope, of course, is one that we use in the classroom. The electron microscope would be one that you might be able to have access to if you go to a university. Now, there's two basic types of electron microscopes. We have something called the scanning electron microscope, and we have the transmission electron microscope. And over here on the right, you can see pictures or micrographs of images that have been taken with each scope. So the SEMs are going to focus a beam of electrons onto the surface of a specimen that is usually coated, believe it or not, with a very thin layer of gold. And these are going to provide 3D images, like you see over here on the right, of whatever we're looking at. So you're not going to be able to see what's inside, but you get a good representation of what's on the outside. Now, the TEMs are going to focus a beam of electrons through a specimen that has been sliced very thin. So this is a preparation technique that we even use with the light microscopes. Now, this is going to be your opportunity to see stuff that is on the inside of that cell. And they're going to be stained with atoms of heavy metals to help, bring out those organelles a bit more clearly. In other words, to provide contrast for those organelles. So TEMs are going to be used mainly to study the internal structures, as we had said, of these cells. Now, sometimes before we can actually use a microscope to look at these different cell parts, what we have to do is we have to prepare the cells. So cell fractionation is going to be very useful for studying cell structure and cell function. So to fractionate a cell, what we're going to end up doing is we're going to end up separating the major organelles from one another. Now, there's going to be a special machine that we're going to use called a centrifuge or an ultracentrifuge, which is just a really fast centrifuge. And this centrifuge is going to spin at various speeds to fractionate the cells into their component parts. So down here you're going to notice that depending on the speed, that's going to determine what we're going to get at the very bottom of the test tube. So if you fractionate at lower speeds, you tend to get the larger, more heavy components at the bottom of your tube. If you fractionate at higher speeds, then it tends to yield the smaller parts of that cell. So cell fractionation is going to enable scientists, primarily the biochemist and the cytologist, to determine the function of the different parts of the cell. So over here on the right, this is just a really quick rundown of how the fractionation process actually works. So you get your sample of tissue cells right here. You're going to homogenize the sample, which basically means put it into a blender and blend it up. You're going to take your homogenate, you're going to expose it to the centrifuge. Then you're going to notice we have different speeds. We have 1,000 times the force of gravity, 20,000 times the force of gravity, 80,000 and 150,000. And then of course we have different times. In other words, how long do we actually spin it? Now, depending on the again, the force and the amount of time, that's going to give us different pellets, is what we call them, at the bottom of the test tube. So if you spin it relatively slow, you're going to get a pellet that's going to be rich in nuclei and cell debris. If you spin it a little bit longer at a little bit more force, you will get a pellet that's going to be rich in mitochondria and chloroplasts if you're looking at a plant cell. If you spin it at 80,000 or 60 minutes, then you're going to have a pellet that's going to be rich in what we call microsomes, and these are going to be plasma membranes, and then of course some of the other organelles that are in there. And then if you spin it really, really quickly for about 3 hours, you might get a pellet at the very bottom that's going to be very rich in ribosomes. So now that we have a good understanding of how we're going to view the cell and how to prepare the cell, we're going to go ahead and begin our discussion by looking at the two primary types of cells. And so when you think about cells, we tend to break them down into two categories. One category is going to be called the prokaryotic cell, and the other category is going to be called the eukaryotic cell. So only organisms of the domain bacteria and archaea, which again are domains that we had looked at back in chapter 1, are going to consist of the prokaryotic cells. Protists, fungi, animals, and all plants are going to consist of eukaryotic cells. So these are going to be the bigger cells. So some basic features of all cells, and so that's going to include both the prokaryotic and the eukaryotic cells, is that all cells do have a plasma membrane or a cell membrane. They do have a semi-fluid substance that's kind of jelly-like in consistency called the cytosol. Sometimes we use the word cytoplasm, where the organelles and other components are going to be found. Both types of cells do carry genes, so they both have chromosomes. It's going to appear a bit different. It's going to be contained a bit differently based on the type of cell, and then both will contain ribosomes. And they both contain ribosomes because the ribosome is going to be the primary site of protein production. So when you look at prokaryotic cells, they're going to be characterized as having, again, they still have DNA, they still have the chromosomes, but they're not going to have a nucleus. So that's something we're kind of accustomed to seeing inside of a cell. They're really, really tiny, so there's really not space for a nucleus. So the DNA tends to be unbound. It's going to be in a region called the nucleoid. They have no membrane-bound organelles, so you're not going to find mitochondria, you're not going to find chloroplasts, you're not going to find Golgi apparatus, some of the parts we're going to look at later. The cytoplasm is going to be bound by a plasma membrane, and it's going to be about 1 to 5 micrometers in diameter. So again, it's going to be a really tiny cell. So down here towards the bottom is just both a diagram and a micrograph of a bacteria. And so some of the parts we looked at was the nucleoid, the ribosomes, which are located here, of course the plasma membrane. We talked about all of these except for a cell wall and a capsule. So when you look at bacteria, they actually have 2 additional layers. They do have a cell wall. It's kind of similar to what you would see in a plant cell, but on top of that, they also have a capsule. Now, sometimes what you'll see with some bacteria is they will have extensions off of the cell. And sometimes they'll have flagella, and then occasionally they'll have maybe these tiny hairs, which you can see right here. They call them fimbria, but to be honest with you, I just call them cilia. But sometimes you'll find those on bacteria cells also. Now, eukaryotic cells, again, are going to be characterized by having the DNA as well, but that DNA is going to be found within a nucleus. And so that's going to be bounded by a membranous nuclear envelope. They are going to have membrane-bound organelles. Cytoplasm is going to be in the region between the plasma membrane, the cell membrane, and the nucleus. Eukaryotic cells are going to be much, much larger than the prokaryotic cells that we looked at in the previous slide. And the plasma membrane, and I think this is similar to what you would see in a bacteria cell, is going to be considered a selective barrier. And that's going to allow sufficient passage of things that the cell needs, like oxygen, could be nutrients, and then of course getting rid of stuff that they don't need, waste, for example. Now, something that I think kind of gets across the idea as to why cells have to stay really small is the idea of metabolism. Now, if you look on the right, I'm guessing this picture probably looks very familiar to you because this is something that we looked at back in Honors Bio. The logistic of carrying out all the different metabolic processes that are necessary to keep the cell alive is very limited, and it's limited based on the size of the cell. So the 2 things we need to look at are surface area, and we need to look at the volume. And what we're going to do is we create a ratio of these 2 values. And so what we notice here is that as the surface area increases, and it increases by a factor of n squared, the volume is also going to increase, but the problem here is that the volume increases at a much faster rate than the surface area. And so it increases at a value of about n cubed. Now, small cells are going to have a greater surface area relative to volume, which basically means that their ratio between those two values is going to be much higher, and you can see that over here on the right. So we have a 1 centimeter, a 2 centimeter, and a 3 centimeter cell. And if you notice, when we calculate the surface area, we have 6, we have 24, and we have 54. Now the volume is going to be calculated as 1, 8, and 27. And so if you notice, when you create a ratio between these two numbers, the surface area to volume ratio for this very tiny cell is 6 to 1, or basically just 6. The one that's gotten a little bit bigger, its surface area to volume ratio is about 3. But then when you look at the really large cell at about 3 centimeters, now you notice it's at about 2. So if you notice, this ratio is getting smaller and smaller and smaller. And what that means to us is, in regards to cells and how they metabolize, is that the idea is we can't get enough nutrients into the cell or enough waste out of the cell to be able to make sure the cell survives. It's much better to have a much smaller cell so there's not as much distance to travel for these two things, and that's represented by the surface area to volume ratio. So you always want a big number here. Now there's lots of different ways that cells have actually remedied this. So it's actually allowed these cells to get bigger. Some cells are going to have different shapes. For example, you might have a cell that's a bit more elongated, and when that happens, what that does is it tends to increase the surface area to help accommodate that increased volume. Sometimes you'll have cells that won't be, I guess you would say, typically shaped. In other words, you won't see a circle, you won't see an oval, but they kind of look something like maybe this. And again, what they're doing here is they're doing their best job at trying to increase that surface area to accommodate that increase in volume as the cell gets larger. So there's lots of different ways out there that cells have been able to get bigger to be able to sort of make sure that this number stays as large as it possibly can. So the very first cell part that we're going to focus on is going to be the nucleus. And so if you notice up here, it says the eukaryotic cell's genetic instructions are going to be housed in a nucleus, and actually the information is going to be carried out by the ribosomes. So the nucleus is going to contain most of the genes, which of course contains the DNA in the eukaryotic cell. Believe it or not just in the nucleus, but there actually is some DNA or genes that are going to be found in the mitochondria and the chloroplasts. So these are other organelles or other cell parts you would find within the much larger cell. Now ribosomes are going to use this information from the DNA, as I had said, to synthesize or make proteins. Now the nuclear envelope, sometimes called the nuclear membrane, is going to enclose the nucleus, and that's going to separate it from the cytoplasm. So the nuclear membrane is considered a double membrane, and each membrane is going to consist of a lipid bilayer, so very similar to what we have seen in the cell membrane. Now this membrane is going to have various holes or pores, and the purpose of the pores is to regulate the entry and the exit of molecules from the nucleus. Now the shape of the nucleus is going to be maintained by a region called the nuclear, I think it's a lamina, which is going to be composed of protein, and that's going to lie right underneath the nuclear membrane. So in the nucleus, the DNA and the protein are going to form a genetic material. It's called chromatin. So it's sort of a combination between those two, a nucleic acid and a protein. The chromatin is going to condense to form discrete chromosomes. Now it's only going to it's only going to condense during cell division. Other than that, it's going to be sort of scattered throughout the nucleus as chromatin. Now the nucleolus is going to be located within the nucleus, and it's going to be the site of ribosomal RNA synthesis. So we're going to be producing RNA within that nucleolus. Now it says proteins with ribosomal RNA form large and small ribosomal subunits. And so this statement right here is basically telling you that these two things combined together are going to create the ribosomes. They're going to be sent out into the cytoplasm. And they're going to exit the nucleus through the pores, and they're going to be assembled as a ribosome. So over here on the right, this gives you an idea of the nucleus found within the cell. And if you notice, the nucleolus is going to be right here. The chromatin is going to be this material that you see scattered through here. So it's not dividing, so it's going to be scattered throughout the nucleus. Over here again you see some micrographs just to give you a more real idea as to what these parts look like. So we have the nuclear pores, which again are going to regulate things going in and out. You can see the nuclear membrane right through here, and this kind of gives you an even closer view of those pores. And down here you can see the pores that kind of look like a little flower, sort of dotting the nuclear membrane. And this actually gives you a good idea of what that nuclear lamina looks like. And this is a TEM micrograph of that material. And again, that's going to lie right underneath the membrane to help support or give form to that nucleus. Now the ribosomes, as we had said, are going to be considered protein factories. So they're going to be there to make out protein. They're going to be there to make protein. They're going to carry out protein synthesis, and this is going to happen in two different locations. One of them is going to be in the cytosol. Now remember, cytosol is the same thing as as cytoplasm, and this is going to contain the free ribosomes. On the outside of the endoplasmic reticulum is going to be another site where you would find ribosomes, and these are considered bound ribosomes. Now down here towards the bottom, as we had said in the previous slide, when you create a ribosome, you have two subunits. One is a large subunit and one is a small subunit. And these subunits are created inside of the nucleus by the nucleolus. And when these subunits leave the nucleus, they're going to be assembled into a ribosome. And so over here on the left, you can see the cytosol, which again is the cytoplasm right through here. You can see the ER, which are these channels, and then these free ribosomes are sort of floating throughout the cytoplasm. But these right here are considered our bound ribosomes, so they're going to be attached directly to this ER. So then what we do is we look at the other organelles, but what we do is we actually put them into a group, and we call this the endomembrane system. And this inside membrane system is going to include the nuclear membrane or envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, the vacuole, and then of course the cell membrane. It says these components are either continuous or they could be connected via transfer vesicles. In other words, all of these different parts that you see listed here are actually connected to each other in some way. So either continuously by their membranes or by the vesicles that will be created to maybe transport substances or materials back and forth between these different parts. So the endoplasmic reticulum, sometimes called the ER, is going to account for more than half of the total membrane in any eukaryotic cell. It is going to be a network of membranous tubules and sacs called cisternae, with the internal space called the ER lumen or cisternal space. It is continuous with the nuclear envelope. Now there are two distinct regions of ER. One of those is called the smooth ER. The second is the rough ER. So the smooth ER is going to be identified by actually lacking ribosomes. Now the smooth ER is going to be responsible for synthesizing steroids, primarily sex hormones and adrenal hormones, cortisol for example. Metabolize carbohydrates, in other words create carbohydrates. It's going to be there to detoxify poisons. Our liver cells do a really good job at that for us. And to store calcium. Now this calcium is important because whenever our muscle tissues need to work, the calcium actually acts as a trigger to make sure that that muscle will contract. Now, as we had said, there's a second type of ER which is called the rough ER, and to identify this one from your smooth is pretty easy because the ribosomes are going to stud the surface of this type of ER. And again, these bound ribosomes are going to be there to secrete protein, more specifically the glycoproteins you would find inside of the cell. Now we use the word glyco to sort of hint that this is a combination between carbohydrates and protein, so they've been kind of bound together. And it's also going to be used to distribute different materials throughout the cell. So it's going to form what we call transport vesicles, sort of like little sacs, and within these sacs are going to be proteins, and these proteins will be moved to different cell parts. So here is just a diagram that's representing what that endoplasmic reticulum would look like. And as we have done in the previous slides, here's a micrograph of this. So if you notice the nuclear envelope, which is going to be right through here, you can see the rough ER right through here, which is identified by these ribosomes that stud the surface. And then you have something called smooth ER right on the outside. and again you can see the lack of ribosomes. Now down here you can see the cisternae, which is going to be the membrane that you see through here, and we have something called the ER lumen. Now what they refer to there is just simply the space in between the folds of the ER. So down here, again, as we had said, sometimes what you'll notice is on the ER, if products, for example, like the glycoproteins or maybe other proteins are being produced, there's going to be something called a transport vesicle, and that's going to move this material away from the ER. So down here, you can see the two different types of ER being represented. We've already looked at this one in a previous slide. This is the rough ER, and it's identified by these ribosomes. Then over here on the left, you can see the smooth ER, which is going to lack these ribosomes. So the Golgi apparatus is the next cell part that we're going to look at, and this is going to be primarily used for shipping and receiving materials within the cell. So the Golgi apparatus is going to consist of flattened membranes called cisternae, and these have polarity, and polarity basically just means difference. It's kind of like what we had looked at with the water molecule. Back when we looked at that, I can't remember if it was chapter 2 or 3, the idea was that if you look at a water molecule, it has a slightly positive end and a slightly negative end. Well, in this case, when you look at this particular organelle, it has what we call a cis phase, which is the receiving end of the Golgi apparatus, and a trans phase, which is going to be the shipping side. So the Golgi apparatus is going to function to modify the products of the ER as they pass from the cis to the trans region of the Golgi apparatus. It's going to be there to manufacture certain macromolecules, for example, pectins. If you've never heard of a pectin before, it's going to be a type of actually a complex carb that's going to be created by plants, and it's going to be sort of embedded with the cellulose within the cell wall of the plant. So it actually acts to provide structure for the plant cell or the plant itself. It's going to be there also to sort and to package materials into transport vesicles from the trans phase. Now if you look down here towards the bottom, again we had said we have a cis phase and we have a trans phase to this organelle. And we might have materials that are coming in maybe from the ER, for example, and they're going to encounter this cis phase. Well, as they make their way in, again, different things could be happening. We could have modifications, we can have sorting, and then of course we might have packaging, and if that packaging occurs, then we're going to get these little transport vesicles that you see right through here. Again, this is the shipping side, which is going to allow this material to be passed on to different parts of the cell. Over here on the right, you can see, again, a micrograph of what this structure would look like. And over here, we're going to have the cis side, which again is the receiving side. And down here, you can sort of identify it by these little sacs. This is going to be the trans phase or trans side, where these are going to be sort of packaged up and sent off to different parts of the cell. Now the next one is going to be the lysosome, and sometimes when you think about the lysosome, you think about the digester of the cell. A lysosome is a membranous sac of hydrolytic enzymes, and so these enzymes are going to be responsible for digesting different macromolecules within the cell. The lysosomal membranes, in other words, that create this structure, and the hydrolytic enzymes are going to be made by the rough ER and then further processed by the Golgi apparatus. So you have two structures here that are working together to create this organelle. Now there are some single-cell creatures such as amoebas which are going to use these lysosomes specifically in a process called phagocytosis. Now this is going to result in the formation of something called a food vacuole. And that food vacuole is going to be used to help digest the food the single-celled organism has taken in. A lysosome will fuse with this food vacuole, and again, as we had said, is going to digest the molecules that have been contained within that food vacuole. Another good example of phagocytosis within our bodies is the white blood cells that we have that are being used to defend our body. And so they use the same process that would be used by the amoebas to basically encounter, engulf any type of foreign invaders, and then of course the lysosome will be there to destroy it. Now lysosomes are also going to use enzymes to recycle the cell's own organelles because they don't last forever. And so when they get older, when they can't do their job as effectively as they used to, the lysosome is going to come by and it's going to engulf them and basically destroy them, and new organelles are going to take their place. And this is a process called autophagy. Now down here towards the bottom again is a micrograph and a diagram of what's going on. So in this case we have phagocytosis, which is simply the taking in of a food particle, and again, food vacuole, a sort of fusion that's going to take place between the lysosome and the food vacuole, and then of course digestion is going to occur. Over here on the right, this is going to be autophagy, and this is when we have some sort of, I don't know, sometimes they refer to them as dead and dying organelles. In other words, organelles that aren't doing what they're supposed to. Lysosome detects that. Again, it's going to be engulfed within a vacuole, and then that fusion is going to occur, and the destruction is going to happen with that organelle. So vacuoles is, again, another pretty, I would say, obvious organelle or structure that you would find within a cell. And you're going to find them both in plant and animal cells. They're going to look a little bit different from each other in each of these, but pretty much they perform the same function. There are 3 different types of vacuoles that you see on the screen. One of them is called a food vacuole, and this is one we had mentioned in the previous slide. And again, these are going to be formed during phagocytosis. Then we have one that a lot of single-cell creatures will use, like freshwater protists, called a contractile vacuole. And these are used to pump excess water out of the cells. And the one down here towards the bottom is one we're probably most familiar with, and this is the central vacuole that you would find in many mature plant cells. And these are going to be there to hold organic compounds and water within the cell. Now sometimes, depending on the type of plant we're looking at, these central vacuoles could also possibly contain pigments. These pigments are going to be used to give the plant color, and sometimes they will actually contain poisonous compounds. And the reason for that is there are some plants out there that are toxic, and so when they're consumed by various predators, sometimes that's going to give a clue to the predator, don't eat this type of plant again. So again, these central vacuoles are there to hold things that the cell might need, but again, sometimes they're useful outside of the cell as well to help protect that organism. So another cell structure that we often are familiar with is something called the mitochondria. Now, some people would call this the powerhouse of the cell. Now, sometimes people think that the mitochondrion is only found in animal cells, and that's definitely not the case. You will find mitochondrion in both plants and animals. Now, when you look at the structure, they have a smooth outer membrane, which you can see right through here. This is considered the smooth outer membrane, and they have an inner membrane that is going to be folded. And so you notice right through here, this is the folded part of the membrane, and that's going to be called the cristae. Now, the inner membrane is going to create 2 compartments, an intermembrane space and a mitochondrial matrix. And so the cristae is going to, as I had said, be identified by the folds, and the matrix is going to be the area between the folds right down here. Now, some metabolic steps of cellular respiration, which we will get to in later chapters, are going to be catalyzed or encouraged in the matrix of the mitochondrion. Now, the cristae are going to present a very large surface area for enzymes that are going to be used to synthesize ATP. And if you remember, this is the powerhouse of the cell, so it's going to be used to make this molecule to help to power various other cell processes. Now, if we're looking at a plant cell, then we're probably going to be able to see a structure called chloroplasts, and the chloroplast is going to belong to a family of organelles called plastids. Now, also included within this group called plastids, we have something called amyloplasts, and these are going to be used to store starch, and chromoplasts, which are going to be used to give fruit their color. Now, a chloroplast is going to contain the green pigment chlorophyll, and there's different types of chlorophyll. Remember back in honors we talked about chlorophyll A and chlorophyll B, and as well as other enzymes. And these are going to function to make sure that photosynthesis occurs within that plant cell. Now, chloroplasts are going to be found in the leaves and other green organs of the plant, and of course you're going to find chloroplasts in single-celled algae. Now, a chloroplast actually is made up of 2 different structures. One of them is called the thylakoid, and the other is called the stroma. Now, there is a third part over here on the right, but this is actually going to be a collection of thylakoids. Now, a thylakoid, you can see down here towards the bottom, are going to be these very thin green discs. Now, when they're stacked up on top of each other, that's when we're going to call them a granum. Now, these thylakoids are going to be there to take in the light that the plant would be exposed to. And so it's going to contain photosynthetic enzymes that are going to help to transport that energy to the various parts that are necessary to make sure photosynthesis occurs. Now, the stroma, this is going to be the internal fluid of the chloroplast, is going to be found in between those stacks of thylakoids, and so in between those grana. And these could contain DNA, as we had said in an earlier slide. Mitochondria and chloroplasts have their own DNA, could contain ribosomes, and definitely does contain enzymes to assist in the process of photosynthesis. Now, for this screencast, the very last cell part that we're going to look at is something called a peroxisome. These aren't mentioned too often, but they are pretty important to the cell because a peroxisome are going to be a very specialized type of organelle that has metabolic compartments that are going to be bound by actually a single membrane in this case. And their purpose is to transfer oxygen from various substrates. There's going to be a lot of involvement when it comes down to the chemical reactions occurring within the cell. Peroxisomes are going to produce something called hydrogen peroxide. So you can see where that second part of that word comes from. And it's going to be used to convert this hydrogen peroxide into water. And they do that because hydrogen peroxide is actually a toxic substance within the cell. So we need to change it into something that is less toxic. Oxygen is going to be used to break down the different types of molecules, such as fatty acids. So that's one of the purposes of a peroxisome. So they can be transferred to the mitochondrion and actually be used as fuel. Now remember, fats, lipids, are a good source of long-term energy. So that's going to be the purpose here of this peroxisome, is to help out with getting that fuel to the mitochondrion. Now some will be used to detoxify alcohol and other harmful substances in the liver. So they actually have a second function as well. Now, as I had said, the hydrogen peroxide is considered toxic, so there is also an enzyme to break it down into, as I had said, water and oxygen, which are two things that our cells definitely need. So it's a really important organelle within our cells to help to detoxify or change things that are probably not too good for us into things that are considered safe. Now down here towards the bottom again, you can see another micrograph. You can see a chloroplast right through here. You can see a mitochondrion off to the right. Then of course you can see this structure here, which is going to be the peroxisome. Alright, so that's going to finish up this very first screencast for Chapter 6. As I said, we're going to break it down into two. Please make sure that you have answered the questions that are associated with this screencast.